the immortal jellyfish. Every living thing on Earth follows the same contract. You are born, you grow, you age, you die. Evolution wrote that contract 4 billion years ago, and nothing has ever broken it, until a 2-millimeter blob of translucent tissue drifting in the warm water off the coast of Portofino, Italy, decided the terms were negotiable. Turritopsis dohrnii was first described by the marine biologist H. Schneider in 1883, collected from the Mediterranean and filed away as unremarkable. For over a century, it sat in the literature as just another small jellyfish. Then in 1996, biologists Christian Sommer and Giorgio Bavistrello watched one do something that had no name in biology. A mature adult, fully sexually developed, reversed, not died, not reproduced, rewound. Its cells reverted to their embryonic state, and it began life again from the beginning, as if the previous version of itself had never happened. The observation was published by Pirino and colleagues in Biological Bulletin that same year, and the quiet shock it sent through the biology community has never fully resolved. The process is called transdifferentiation. When the jellyfish is stressed, injured, or simply old, its differentiated adult cells, muscle cells, nerve cells, fully specialized tissue revert to undifferentiated stem cells. The organism does not die. It resets, not a pause, not dormancy, a complete biological rewind to the beginning of the developmental sequence. The evidence for this is now extensive. The 1996 Pirino paper in Biological Bulletin was the first peer-reviewed confirmation of complete biological reversal in a metazoan, meaning an animal with differentiated tissues, and it established transdifferentiation as the operating mechanism beyond reasonable dispute. The genes activated during this reversal, specifically OCT4, SOX2, and KLF4, are the same genes that scientists use to artificially reprogram human cells into stem cells in regenerative medicine experiments. Cancer researchers have spent decades trying to replicate this process in laboratory settings. The jellyfish does it in response to stress. In 2009, Maria Pia Miglietta of Texas A&M University confirmed that the species had been silently colonizing every major ocean by hitchhiking in ship ballast water, potentially producing billions of genetically identical immortal individuals without anyone noticing. The animal measures approximately 4.5 millimeters at maximum maturity, smaller than a human fingernail, which explains how a global population expansion went undetected for decades. At Kyoto University, Shin Kubota has maintained a laboratory colony for over 20 years, documenting more than 100 complete rejuvenation cycles in individual specimens, with no upper limit yet established. Individual animals under his observation have reversed the aging process as many as 10 consecutive times, with no measurable physiological degradation detected between cycles. Biologists classify transdifferentiation as a stress response, an emergency survival mechanism shaped by extreme environmental pressure. When resources collapse or damage exceeds the body's repair capacity, reverting to an earlier developmental stage may be energetically cheaper than dying. In this view, Turritopsis dohrnii is a very extreme form of dormancy, not a biological exception to aging, but a workaround that sidesteps the conditions that normally trigger it. It is, the mainstream argument goes, an elegant adaptation, not a miracle. But dormancy is a pause. This is a rewind. The jellyfish does not suspend its cellular processes, it reverses them completely. And the genes it uses to do so are not unique to jellyfish. They are present in human DNA right now, largely switched off, their full function still unmapped by science. If transdifferentiation is purely a stress response, why does it produce a fully functional, reproductively capable juvenile rather than simply stabilizing the adult? Some researchers have begun asking whether this mechanism is not a survival trick at all, but something closer to a biological clock that was never meant to have a terminal setting. Is it possible that the genes governing reversal in jellyfish are the dormant version of the same program sitting inside every cell in the human body, waiting for a signal we have not yet identified? And if so, what switched them off and when? We may never know. What we do know is that somewhere in every ocean right now, an immortal animal smaller than your fingernail is quietly beginning again. From an animal that refuses to die, we cross the Pacific to a creature that sees a version of reality the rest of us are physically incapable of imagining. The mantis shrimp. You think you see the ocean. You see blue water, green coral, silver fish. The mantis shrimp looks at the same reef and perceives a version of reality your brain is physically incapable of processing. Then it kills something faster than you can blink, using a strike that briefly generates the temperature of the surface of the sun. Coastal fishermen have known about mantis shrimp for centuries. In some communities, they are called thumb splitters, a name earned by their ability to shatter aquarium glass and split open the fingers of handlers who underestimate them. But the full extent of their visual and mechanical capabilities was not systematically understood until the 1990s, when Justin Marshall of the University of Queensland and Thomas Cronin of the University of Maryland began publishing work that would fundamentally revise what biologists thought they knew about color perception in the animal kingdom. The mantis shrimp possesses 16 types of photoreceptor cells. Humans have 3. The butterfly, long considered the champion of color vision among animals, has 5. But the mantis shrimp does not simply see more colors. It processes color through a mechanism so architecturally different from vertebrate vision that neuroscientists are still building models adequate to describe it. And when it is finished seeing, it strikes. Marshall and Cronin's 2001 paper in Nature confirmed that the mantis shrimp processes color locally at the receptor level rather than in the brain. This is the precise inverse of all known vertebrate visual architecture, in which raw color signals are transmitted to the brain and mixed there into perceived color. The mantis shrimp's 16 photoreceptor classes span wavelengths from deep ultraviolet at 300 nanometers to far red at 720 nanometers, a visual range twice as broad as human vision, covering portions of the electromagnetic spectrum we cannot perceive at any intensity whatsoever. The striking appendage, called the dactyl club, accelerates at 10,400 times the force of gravity, faster than a .22 caliber bullet, delivering impact forces of 1,500 newtons from an animal weighing roughly 70 grams. On impact, cavitation bubbles form and collapse at temperatures reaching approximately 8,000 Kelvin, comparable to the surface of the sun, arriving as a second strike milliseconds after the physical blow. David Casailas of UC Irvine documented the club's internal architecture in a 2012 paper in Science and found a helicoidal fiber structure that dissipates impact energy so efficiently that engineers are now replicating it in aircraft body panels and military body armor. Beyond all of this, mantis shrimp can recognize individual neighbors and adjust territorial behavior based on that recognition, a cognitive capacity previously considered impossible in crustaceans. The order Stomatopoda, which contains them, diverged from other crustaceans approximately 400 million years ago, 170 million years before the first dinosaur walked the Earth. Biologists argue that the 16 photoreceptor system evolved as a speed-optimized color identification shortcut. Rather than mixing color signals in the brain, the mantis shrimp identifies discrete spectral flags rapidly, like reading barcodes, allowing fast identification of prey species, threat signals, and mate fluorescence patterns in the visually complex, rapidly changing environment of a coral reef. The dactyl club, in this view, is an efficient predation tool in an environment where hard-shelled prey like crabs, snails, and bivalves are abundant. But the barcode model does not explain why an animal needs to read barcodes in ultraviolet and far infrared simultaneously, wavelength ranges where no known prey species and no known reef predator emits a detectable signal. And it does not explain why the club's internal structure is more mechanically sophisticated than anything human materials engineering currently produces for equivalent impact loads. Some materials scientists have noted that the helicoidal fiber geometry of the dactyl club appears in scattered locations across nature, in bone, in insect cuticle, in fish scales, suggesting it may represent a convergently discovered optimal solution to a fundamental physics problem. If a shrimp solved it 400 million years ago, the question, some researchers suggest, is not just why it has such a weapon. It is what exactly the weapon was originally built to fight, and whether whatever that was is still somewhere in the ocean below where we have looked. From a creature that outperforms our senses and our engineering, we descend into deeper water, to an animal that outperforms both by being, technically, not one animal at all. The siphonophore. The longest animal on Earth is not a whale. It is not a whale shark. It is a creature that most marine biologists are not entirely sure qualifies as a single animal, a living rope 50 meters long, hunting without a brain, made entirely of individuals that cannot survive on their own. Siphonophores have been known since the 19th century, but their true scale was invisible to science for most of that time. Every net that attempted to collect them destroyed them on the way up. It was only when remotely operated vehicle technology allowed undisturbed observation at depth that researchers understood what was actually down there. In 2005, scientists at the Monterey Bay Aquarium Research Institute documented a specimen of Praia dubia in Monterey Canyon, measuring between 40 and 50 meters, the longest animal reliably measured in the history of biology. It had been there all along. A siphonophore is a colony of genetically identical zooids, individual organisms, each so highly specialized that it performs only one biological function and cannot survive independently. Some provide propulsion, some capture prey, some handle reproduction, some form the gas-filled float that keeps the colony positioned in the water column. Together they form what functions as a single predatory animal, coordinated, responsive, and lethal, across a body the length of half a football field. The 2005 MBARI documentation of Praia dubia at 40 to 50 meters exceeded the blue whale's maximum confirmed length of approximately 30 meters and established the siphonophore as the longest animal on record. The hunting net of tentacles can extend over 30 meters horizontally, equipped with pneumatocysts that fire on contact, a coordinated trap covering an area larger than a tennis court. In 2020, a genomic study published in Nature Ecology and Evolution by Stefan Siebert of UC Berkeley confirmed that siphonophore zooids share identical DNA but express entirely different gene sets, meaning the same genome produces radically different body forms depending on where in the colony a given zooid is positioned. Signal transmission across the colony has been observed in coordinated waves with no identifiable central point of origin, and the mechanism driving that coordination is not understood. Some species produce bioluminescent displays along the full length of their bodies. 50 meters of synchronized light pulses in the deep ocean, the function of which remains completely unknown. According to some estimates, siphonophores consume more prey annually than any other group of marine predators, constituting a significant and largely unquantified fraction of total deep ocean biomass. Biologists classify siphonophores as colonial organisms, analogous in principle to a coral reef but mobile. Coordination between zooids, in this view, emerges from simple chemical and electrical signals passed between adjacent units, no central processor required. Division of labor is energetically efficient. A colony in which every member performs one task with perfect specialization outcompetes generalist organisms in resource-scarce environments. The structure is complex, but the underlying principle, according to mainstream biology, is not. But a coral reef does not hunt. It does not pursue prey. It does not make collective real-time decisions across 50 meters of body. The siphonophore does all of these things without a brain, and the mechanism by which coordinated predatory behavior emerges from signal passing between adjacent zooids has never been fully characterized. The siphonophore raises a question that philosophers of biology have not resolved. What is an individual? If every zooid is genetically identical and none can survive alone, is the colony the individual, or is it something else, a distributed organism achieving coordinated behavior through a mechanism our science does not yet have adequate vocabulary to describe? Some researchers have begun suggesting that living siphonophores may offer a model for understanding how the first multicellular animals organized a glimpse, still alive and hunting in the deep Pacific, of the transition from single cell to coordinated body. Whether that interpretation holds is supposedly still open. What is not open is that a 50-meter animal has been hunting in Monterey Canyon for longer than we can calculate, and we found it only when we finally built machines that could go and look. From a living colony hunting in the dark present, we move north into frozen water to something that has been circling in that darkness since before the Renaissance, the Greenland shark. Somewhere in the black water beneath the Arctic ice, there is a shark that was alive when Michelangelo was painting the Sistine Chapel. It has been moving through the dark at approximately 1 meter per hour for over 500 years. It is not yet old enough to reproduce. The extreme longevity of Somniosis microcephalus was confirmed in a landmark 2016 study by Julius Nielsen of the University of Copenhagen, published in Science. Nielsen's team used radiocarbon dating of the crystalline lens nucleus, tissue that forms before birth and never regenerates, to establish the ages of 28 individual specimens. The results did not simply set a new record for vertebrate longevity. They made the previous record look like a rounding error. The oldest individual in Nielsen's sample was dated to between 1500 and 1744, older than the United States, older than the telescope, older than the printing press. The dating method works by using atmospheric carbon-14 spikes from Cold War nuclear testing as a calibration point. Sharks born after 1955 carry the spike in their lens tissue. Sharks born before do not. This allows stratification of the entire sample population into pre- and post-nuclear cohorts with reasonable precision. The species grows approximately 1 centimeter per year, which means a 5-meter adult is, at minimum, 500 years old. Sexual maturity arrives at approximately 150 years, meaning Greenland sharks spend longer as juveniles than the entire recorded history of the United States as a nation. The flesh contains high concentrations of trimethylamine oxide, a cryoprotectant that prevents cellular ice crystal formation and makes the meat toxic when fresh. The traditional Icelandic preparation called hakarl requires 6 months of fermentation to render it safe to eat. Approximately 90% of examined specimens are infected with Omatocoida elongata, a parasitic copepod that attaches to the cornea and causes near-total blindness, yet the sharks remain effective predators, with documented stomach contents including seals, large fish, and in one case the complete remains of a polar bear. Body temperature matches ambient water temperature near 0 degrees Celsius, meaning the shark's biochemistry operates on metabolic timescales unlike those of any other vertebrate. The metabolic rate hypothesis offers an explanation. At near freezing temperatures, biochemical processes slow so dramatically that cellular aging, driven by metabolic byproducts like reactive oxygen species, slows proportionally. Low metabolism means low damage accumulation over time. The Greenland shark is, in this view, a very cold and very slow animal that has not yet had sufficient reason to die. The explanation is reasonable. It is consistent with the known biology of cold water species, but metabolic slowdown explains gradual aging. It does not explain the apparent near absence of it, and it does not explain how an effectively blind predator moving at less than 1 mile per hour hunts and kills fast-moving marine mammals regularly enough to sustain a body the size of a great white shark. Julius Nilsson himself has noted that temperature alone may not fully account for his specimen's longevity, and that there may be active biological suppression of cellular aging that has not yet been isolated. The trimethylamine oxide that makes the flesh toxic may play a role in cellular stabilization beyond its function as a cryoprotectant. An animal that reaches reproductive maturity at 150 years did not arrive at that timeline by accident. Something, according to some researchers, selected specifically for extreme longevity. The question they are beginning to ask, quietly, is what survival pressure required it, and whether whatever that pressure was is still present somewhere in the Arctic dark, keeping the slowest predator on Earth moving forward. The Greenland shark navigates centuries in near total darkness without eyes. The next animal solved the problem of darkness differently by replacing its skull with a window. The barreleye fish. For 65 years, scientists held the head of this fish in their hands and did not understand what they were looking at. The dome they assumed was a rigid skull was actually a window, and the two green spheres floating inside it, the ones that every diagram and every textbook labeled as eyes for 6 decades, were not eyes at all. Macropinnomicrostoma was scientifically described in 1939, but every specimen ever examined before 2004 had been retrieved by net, a process that ruptures the transparent fluid-filled dome covering the head. Each time, researchers opened their trawl and found a flattened, damaged structure where the dome had been and drew their conclusions from the wreckage. In 2004, Bruce Robison and Kim Reisenbichler of the Monterey Bay Aquarium Research Institute descended into Monterey Bay with a remotely operated vehicle and filmed the animal alive and intact for the first time. What they saw required a complete revision of the species description. The entire published understanding of the animal's anatomy, accumulated over 65 years of study, was wrong. The transparent dome is composed of fibrous tissue stretched over a cartilaginous rim. Not a skull, not a membrane. A unique biological structure confirmed by histological analysis following the 2004 observation, found in no other known vertebrate anywhere in the animal kingdom. The actual eyes of the barreleye fish are the two tubular green structures visible through the dome, each with an aperture of approximately 1 centimeter, optimized for collecting faint downwelling surface light at depths of 800 meters, where prey can be detected as silhouettes against the dim glow from above. These eyes can rotate 90 degrees inside the transparent dome, from upward facing when the fish is scanning for prey to forward facing when it has located a target and is moving to capture it. This rotation was first documented in the MBIRI footage and published by Robison and Reisenbichler in Copaya in 2008. What every diagram previously labeled as eyes were the fish's olfactory organs. Large, flat, transparent pectoral fins allow the animal to hover motionlessly in the water column for extended periods while scanning. Stomach content analysis suggests the barreleye regularly steals food directly from siphonophore tentacles, approaching the stinging threads, rotating its eyes forward at the moment of extraction, apparently using the dome as protection from nematocyst discharge. Individuals have been directly observed hovering inside the bells of large jellyfish, apparently using them as shelter. The confirmed depth range runs from 400 to 2,500 meters, with most active foraging behavior recorded between 600 and 800 meters. The tubular eye design is a well-understood adaptation for extreme, low-light conditions. Upward-pointing eyes with wide apertures maximize photon collection from above, where prey silhouettes are most detectable against faint surface light. The transparent dome, biologists argue, evolved as a protective barrier against nematocysts, a specialized structure solving a specific predation and defense problem simultaneously. But no other vertebrate at any depth evolved a transparent head to solve the low-light problem. Hundreds of species share the same depth range. They evolved larger eyes, more sensitive retinas, bioluminescent lures, wider gapes. Only this one replaced its skull with tissue you can see through. The barreleye was systematically misidentified for 65 years. It was not hiding in the abyss, it was swimming in Monterey Bay, one of the most heavily studied patches of ocean in the world. Researchers were there. They simply did not know what they were looking at. NOAA estimates that more than 80% of the ocean has never been directly observed. The barreleye was found in the fraction that has been. Some researchers have begun asking how many other species are currently described from the wrong version of themselves, their most essential features destroyed before the first scientists could see them intact. We have been misreading the ocean for over a century. The next animal has been waiting in that same darkness for 125 million years, appearing without warning in fishing nets, and even now, after 125 years of scientific records, fewer than 50 individuals have ever been meaningfully studied. The goblin shark. Its jaw is not attached to its skull. When it detects prey, the entire lower face launches forward, outside the head, on a mechanism built from cartilage and elastic ligament. No other vertebrate on Earth does this. In 125 years of scientific records, fewer than 50 specimens have provided meaningful data. We have almost no idea how this animal lives. The first specimen was acquired by British naturalist Alan Austin from a Japanese fisherman in Tokyo Bay in 1898 and formally described by David Starr Jordan as Mitsukurina austoni. The jaw projection mechanism, suspected from the anatomy but never directly observed, was not filmed in action until 2008 when researchers at the Florida Museum of Natural History analyzed high-speed video footage and published the first kinematic description of the strike. The jaw extends to approximately 8.6 to 9.4% of the shark's total body length in under 0.3 of a second, confirmed in analysis by Kazuhiro Nakaya and colleagues in a 2016 study in the Journal of Zoology. The mechanism is powered by elastic recoil from ligaments connecting the jaw to the skull cartilage, analogous to a crossbow rather than muscular contraction, meaning the deployment requires no muscular effort from resting state and generates maximum speed instantaneously. The shark is pink, its skin so thin that subdermal blood vessels are visible through unpigmented tissue, with skin thickness in examined specimens approximately half that of other shark species at equivalent size. The ampullae of Lorenzini, the electroreception organs that detect the bioelectric fields of prey, are proportionally larger in this species than in any other known shark, suggesting sensitivity to electrical fields substantially exceeding that of all other selachimorphs. Maximum confirmed length is approximately 6.2 meters, documented in a Japanese specimen in 2000, with average adults reaching 3 to 4 meters. The confirmed depth range runs from 270 to at least 1,300 meters. The most recent significant specimen was recovered in 2014 from the Gulf of Mexico following deep-water disturbance events associated with offshore drilling activity, suggesting populations exist in regions where they were previously unrecorded. And the entire family Mitsukurinidae now contains exactly one living member. All known relatives are extinct, disappearing from the fossil record approximately 11 million years ago. The goblin shark's specializations are consistent adaptations for an ambush predator in a resource-scarce, lightless environment. The slingshot jaw allows rapid prey capture without pursuit. Thin skin reduces metabolic cost. Enhanced electroreception compensates for the complete absence of visual input. The species appears rarely in scientific records. The mainstream argument holds, not because it is rare in the ocean, but because it occupies depths that are rarely fished or directly observed. But the jaw projection mechanism requires a degree of cartilaginous elasticity so specific that no other lineage in 400 million years of shark evolution independently developed it. Every other deep ambush predator, the anglerfish, the viperfish, the fangtooth, uses suction, wide gape, or bioluminescent lure. Only this animal built a catapult inside its face and kept that design unchanged for 125 million years. Fossil evidence suggests that earlier Mitsukurinids inhabited shallower water. Some paleontologists have proposed that the living goblin shark retreated to depth, not because depth suited it, but because everything in shallow water that could compete with it or threaten it is gone. It may not be a deep-sea specialist by original design. It may be, according to this interpretation, a surface ocean animal that lost the surface ocean and retreated into darkness, still carrying the weapons it built for a world that no longer exists. But whether that world ever comes back, and whether this animal is still waiting for it, we may never know. The goblin shark hides from a world that moved on without it. The next animal lives where almost nothing else can even breathe, in a layer of ocean so stripped of oxygen that most marine life suffocates before it arrives. The vampire squid. There is a band in every ocean where almost nothing survives. Not a depth, not a temperature zone, but a chemical condition, water so depleted of dissolved oxygen that fish suffocate, bacteria struggle, and most marine life simply cannot function. Almost nothing lives there, except one animal that turned the worst conditions on Earth into a private ocean, and did it by surrendering the one thing every predator refuses to give up. Vampirotuthis infernalis was first described in 1903 by the German marine biologist Carl Chun, who named it vampire squid from hell based on its dark coloration and the webbing connecting its arms, which spreads like a cloak when extended. For decades, biologists classified it alternately as a squid and as an octopus, before it was recognized as the sole surviving member of a completely separate cephalopod order, Vampiromorphida, in 1915. It is neither squid nor octopus. It occupies its own evolutionary branch alone. The oxygen minimum zone at the vampire squid's preferred depth contains between 0.5 and 1.5 milliliters of dissolved oxygen per liter, approximately 3 to 6% of surface concentration, conditions lethal to virtually every other cephalopod. The species survives this through two structural adaptations: the highest gill surface area to body mass ratio of any known cephalopod, and a form of hemocyanin, the copper-based oxygen carrier in cephalopod blood, with exceptionally high oxygen affinity, allowing it to extract sufficient oxygen from water that would asphyxiate its relatives. It has no ink sac. This has been confirmed across all examined specimens. In an environment that excludes most visual predators, the defensive ink ejection system common to every other cephalopod was abandoned entirely over evolutionary time. In place of ink, the animal releases a cloud of bioluminescent mucus when threatened, a glowing decoy deployed in permanent darkness. A landmark 2012 study by Hank John Hoving and Bruce Robison of MBARI, published in Proceedings of the Royal Society B, confirmed through stomach content analysis and mucus filament sampling that the vampire squid does not hunt living prey at all. Two retractile filaments extend from between the arms to lengths up to twice the body length and passively collect marine snow, the slow rain of dead organic particles, fecal pellets, and shed mucus drifting down from the surface ocean. When threatened, the animal inverts its webbed arms over its body in the pineapple posture, exposing spiny projections on the undersides of its arms, a defensive display documented across multiple MBARI remotely operated vehicle observation sessions. This is, in the mainstream view, a textbook case of evolutionary regression under relaxed selection pressure. In an environment excluding most predators, ink became unnecessary. In an environment where living prey cannot survive, active hunting became impossible. The animal adapted to consume what the oxygen minimum zone does contain, a slow but reliable supply of detritus, and its anatomy simplified accordingly. What remains is an efficient, low-energy organism occupying a niche no competitor can reach. But evolutionary regression requires millions of years of stable selective conditions. The oxygen minimum zone is not a fixed environment. Its depth and oxygen levels fluctuate with climate and ocean circulation on timescales of decades. An animal that abandoned predation and ink entirely did not hedge. It committed completely to a strategy that works only if specific conditions hold indefinitely. Some researchers have proposed that the ancestor of Vampiroteuthis was not drawn into the oxygen minimum zone by opportunity, but displaced there by competition, that it lost, retreated into a habitat no competitor was equipped to follow it into, and survived by becoming something no other cephalopod was willing to become. Current climate models project expanding ocean deoxygenation over the coming century. The vampire squid's private oxygen desert is supposedly growing. Whether the animal designed for scarcity will inherit an ocean increasingly defined by it is a question some researchers are asking with something between scientific interest and unease. From an animal that retreated into the worst conditions on Earth and found a way to thrive, we crossed to the South Pacific, where in March of 2005, a robot descended to a hydrothermal vent field and found something that appeared to be deliberately farming: the yeti crab. In March of 2005, a deep-sea robot descended to a hydrothermal vent field 2,228 meters beneath the South Pacific and found something that fit no known category: a blind white crustacean covered in what looked like fur. It was a new species, it was a new genus, it was a new family, and it appeared to be growing its own food on its arms. The discovery was made during a NOAA-funded expedition aboard the research vessel Atlantis, operating over the Easter Island microplate. The species was described by Michel Segonzac of Ifremer, France, and colleagues in the Journal of the Oceanographic Society of Japan in 2006. They named it Kiwa hirsuta. Kiwa for the Polynesian goddess of shellfish. Hirsuta from the Latin for hairy. The name captured what you could see without yet explaining it. The fur is not fur, it is a dense mat of filamentous bacteria cultivated on modified bristle structures, called setae, covering the animal's claws and body. The bacteria identified on these structures include both sulfur-oxidizing and methane-oxidizing chemolithotrophs, organisms that extract energy from the chemical emissions of hydrothermal vents rather than from sunlight or organic matter. The crab waves its claws rhythmically in the vent output, rocking side to side, maximizing bacterial exposure to the hydrogen sulfide and methane flowing from the vents. This wave-rocking behavior has been directly correlated with bacterial mat density in observational studies, meaning the behavior actively manages the productivity of the crop. The bacteria are then consumed. A second species, Kiwa porivida, meaning living good in Costa Rican Spanish, was discovered at methane seeps off the coast of Costa Rica in 2011 and described by Andrew Thurber of Oregon State University. It shows more pronounced wave-rocking behavior correlated with direct positioning over methane seep outputs. A third species, Kiwa tyleri, was described in 2015 from hydrothermal vents in Antarctica, meaning the genus has colonized 3 completely separate vent systems across the Pacific and Southern Ocean. Kiwa hirsuta itself is functionally eyeless. Vestigial eye structures are present but non-functional. Navigation, foraging, and predator avoidance operate entirely through chemoreception and mechanoreception. At vent sites, population densities reach hundreds of individuals per square meter, among the densest known aggregations of megafauna anywhere in the deep ocean. The bacterial cultivation behavior is interpreted by biologists as a highly specialized feeding symbiosis selected for by evolution rather than directed by any form of intention. The crab performs stereotyped wave-rocking behavior because individuals who performed it more consistently derived better nutrition and achieved higher reproductive success. The bacteria are maintained as a renewable food resource in an environment where alternatives are scarce and unpredictable. Sophisticated, the mainstream argument holds, but explicable by standard evolutionary pressure. But the distinction between stereotype behavior selected for farming efficiency and farming narrows considerably when 3 separate species of the same genus in 3 separate ocean basins with no known connection between them all independently arrived at the same bacterial cultivation strategy on the same body structure. We do not know where the Kiwa lineage came from before it was a vent specialist. No shallow water relatives have been identified. No transitional form appears in the fossil record. Some researchers have suggested that Kiwa may have been farming bacteria at hydrothermal vents for millions of years before we found it, and that we found it in 2005 only because we had finally built instruments capable of reaching it. What it was before it descended to the vents and what pressure sent it there is, according to current knowledge, unknown. The yeti crab created a food supply where none should exist. The next animal found a simpler solution to scarcity. It simply stopped needing to eat for years at a time, and the record it holds has never been broken. The giant isopod. It is a pill bug. The same creature you find under garden rocks, curling into a ball when you lift a stone. This one is the size of a football. It lives 2,000 meters underwater. It has not eaten in 5 years and 43 days. And as of the moment its keepers recorded the end of that fast, it was still alive. Bathynomus giganteus was first described by the French zoologist Alphonse Milne Edwards in 1879 from a specimen trawled in the Gulf of Mexico. The species exemplifies deep-sea gigantism, a broadly observed but incompletely explained phenomenon in which certain deep-sea lineages develop dramatically larger body sizes than their shallow water counterparts. The fasting record was not set deliberately. It was set by an individual held at Toba Aquarium in Japan, which refused all food from the 2nd of January 2009 until its death on the 14th of February 2014, 5 years and 43 days. Staff documented the fast throughout. The animal showed no measurable organ decline until its final weeks. The record has never been equaled by any animal in human care. The maximum confirmed length of this species is 76 centimeters, documented in a 2010 specimen. Average adults measure between 19 and 37 centimeters, making them 14 to 37 times the size of the largest known shallow water isopod, a size increase of more than an order of magnitude over their pillbug cousins. The compound eye contains approximately 4,000 individual facets, among the most complex eyes in all crustacea, providing wide-angle, low-light detection across the deep-sea environment. When food is encountered, the stomach capacity relative to body mass is among the highest of any crustacean, consistent with an opportunistic feeder that evolved to consume maximum calories in a single encounter after an extended fast of unpredictable duration. Hemolymph analysis from fresh specimens reveals unique lipid compositions that may function as long-term energy storage molecules, but the complete biochemical mechanism sustaining multi-year fasting has not been isolated or characterized. The species survives pressures exceeding 200 atmospheres, the equivalent of 2,000 meters of water column, without any identifiable specialized pressure adaptation structure in the exoskeleton. Deep-sea gigantism in isopods has been attributed to the inverse relationship between metabolic rate and body size in cold water, reduced predation pressure allowing survival to greater age and mass, and higher dissolved oxygen at depth. But none of these factors individually predicts the order of magnitude size difference observed between this species and its shallow water relatives. Giant isopods are, in the mainstream view, a standard case of deep-sea gigantism with well-proposed contributing mechanisms. Multi-year fasting tolerance is interpreted as adaptation to a genuinely unpredictable food supply, with metabolic rate suppressed to a minimum sustainable floor during food-absent periods. Cold water slows everything. An animal that waits long enough in cold enough water simply requires less. But metabolic suppression explains slower starvation. It does not explain 5 years of maintained organ function in an arthropod. The Toba specimen was not declining slowly toward death for 5 years. It was operating at some reduced but functional level on a metabolic program that has no equivalent in any comparable phylum. The biochemistry of that program has not been found. The giant isopod may contain solutions to metabolic suspension that are qualitatively novel, not extreme versions of known fasting mechanisms, but fundamentally different approaches to cellular energy management under prolonged deprivation. For an animal as large, as accessible, and as easily maintained in captivity as this species, the absence of a characterized fasting mechanism is, according to some researchers, a conspicuous gap in the literature. What Bathynomus giganteus is actually doing to remain alive during those 5 years is technically unknown. It waits in darkness for food that may take years to arrive, sustained by a biochemistry that science has not yet read. The giant isopod waits in silence for whatever comes. The last animal on this list does not wait. It performs, selecting from a repertoire of at least 15 disguises based entirely on who, at this particular moment, is watching. The mimic octopus. It is lying on the sand looking like nothing in particular, a mottled shape against the seafloor of the Flores Sea, off the coast of Sulawesi, Indonesia. A predator approaches. In under a second, the animal transforms. 8 arms flatten, narrow, and trail in the current. It has become a flatfish. The predator passes, the shape relaxes, then it becomes something else, then something else again. It has at least 15 masks. It chooses which one based on which predator is watching, and it has been doing this undetected in heavily dived tropical waters for an unknown period before anyone recognized it for what it was. The Moctopus mimicus was first observed and filmed in 1998 by Indonesian dive instructors Norman and Steen during a recreational dive in the Flores Sea. The footage reached Roger Hanlon of the Marine Biological Laboratory at Woods Hole, who confirmed it as an undescribed species and initiated formal study. The animal was formally described in 2005 by Mark Norman and Darmanto, the name drawn from the Greek thauma, meaning wonder, and the Latin mimicus, meaning imitative. It had been performing in front of human observers for years before science had a name for the performance. The first formal behavioral study, published in 2001 by Norman, Finn, and Tregenza in Proceedings of the Royal Society B, documented 7 distinct impersonation behaviors in a single individual across a 2-hour observation period. The flatfish impersonation requires the animal to flatten all 8 arms bilaterally alongside its body and undulate them simultaneously to produce a fin wave motion, while the mantle is compressed and patterned with the bilateral symmetry of a flounder. This requires independent simultaneous control of 8 separately mobile limbs coordinated into a single, coherent shape. Sea snake impersonation involves extending 2 arms in opposite directions with black and white banding displayed, and critically, this behavior is specifically and preferentially triggered by the approach of damselfish, which are natural prey of sea snakes. This means the octopus is not simply performing a fixed defensive routine. It is selecting a mimicry target based on the identity of the animal watching it. Documented impersonation targets include, at minimum, the flatfish, the banded sea snake, the lionfish, the crinoid, the mantis shrimp, and the jellyfish, each requiring a distinct postural configuration and behavioral routine. The species has no ink sac, confirmed across all examined specimens, meaning it has abandoned the escape option available to every other octopus and relies entirely on performance. Skin chromatophore cells can be individually activated to produce patterns at resolutions below 1 mm, controlled through a distributed peripheral nervous system in the skin itself rather than through central brain commands. And the entire lifespan of Thaumoctopus mimicus is approximately 9 months. All cognitive capacity required to develop, store, and selectively deploy 15 behavioral impersonation routines must develop and operate within less than a year of life. Biologists describe the mimic octopus's behavioral repertoire as an unusually complex extension of the background matching and disruptive coloration capabilities common to cephalopods. Predator-specific mimicry selection, if fully confirmed, would represent sophisticated learned behavior enabled by the octopus's large and distributed nervous system. Remarkable by any measure, but consistent, the mainstream argument holds with the known cognitive range of the cephalopod class. But background matching is passive, selecting a specific behavioral performance from a repertoire of 15 based on identifying which particular predator is currently approaching is not camouflage. It requires an internal representation of self, a model of other species' appearances and behaviors, and a functional mapping between observed threat identity and optimal response. That is not background matching. That is something closer to applied theory of mind, deployed within a 9-month lifespan by an animal with no backbone and a brain roughly the size of a walnut. 2/3 of the octopus's neurons are located in its arms, not in its central brain. Whether that distributed architecture processes and stores behavioral information through mechanisms that our current neuroscience models do not yet describe is, according to some researchers, a genuinely open question. The mimic octopus was unknown to science until 1998. It lives in shallow, heavily trafficked, well-documented tropical waters. It had been performing, in full view, for an unknown period before anyone understood what they were watching. The question of what else is currently performing in water we visit regularly, unrecognized because we do not yet have the framework to recognize it, is not a comfortable one. These are not anomalies. They are not edge cases at the margins of evolution, statistical noise in the record of life on Earth. They are the ocean stating, repeatedly and with specificity, that the rules we wrote about what biology is allowed to do are incomplete. An animal that reverses aging. A crustacean that perceives wavelengths of light that have no name in any human language. A colony 50 meters long that hunts without a brain. A vertebrate born before Shakespeare still circling in the Arctic dark, not yet old enough to reproduce. A fish whose skull is a window. A shark whose jaw is a catapult, the last survivor of a lineage that lost the surface ocean 125 million years ago. A cephalopod that gave up hunting to survive in dead water. A crustacean farming bacteria on its own arms at the bottom of the South Pacific. An arthropod that maintains organ function for 5 years without food on biochemistry we have not characterized. An invertebrate with a 9-month lifespan that selects its disguise based on who is watching. None of these were predicted. All of them were found, and all of them were found in the roughly 20% of the ocean that has been directly observed. Mainstream biology has an explanation for each animal on this list. Transdifferentiation as stress response. Visual barcode reading as efficient color identification. Colonial coordination as emergent behavior from simple signal passing. Longevity as metabolic consequence of cold water. Each explanation is reasonable. Each explanation is also incomplete in at least one specific documented way that the researchers who study these animals will acknowledge if you ask them directly. Some scientists have begun suggesting, carefully, that what we have described represents a fraction of what exists, and that the mechanisms we have identified explain the surface of what these animals are doing without fully accounting for how they do it. Not a conspiracy of suppression. A straightforward acknowledgement that 80% of the ocean has never been directly observed, and that the 20% we have seen keeps producing things we did not expect. The barreleye fish swam past researchers for 65 years before anyone filmed it intact. The mimic octopus performed in front of divers for an unknown period before anyone recognized the performance. The yeti crab farmed bacteria at 2,228 meters until 2005, when we finally built a machine that could reach it. The ocean is not hiding these things from us. We are simply still looking at it with incomplete instruments and a confidence in our understanding that the evidence, repeatedly, does not fully support. Most people look at the ocean and see a backdrop. You watch this far because you understand it is a record and that most of it has not been read. The animals that exist in the 80% we have not seen are, statistically, stranger than anything on this list. That thought is either exciting or unsettling, depending on who you are. This channel exists for the people who find it exciting. Subscribe and stay in the archive. Then go to the comments and tell us which animal on this list breaks your model of what life is supposed to be capable of. One animal, one argument. Let's see the reasoning. The ocean has been producing life for 3.8 billion years. We have been systematically studying it for approximately 200. In that time, we have directly observed less than a fifth of it. Everything on this list was found in that fifth. Somewhere in the Mediterranean right now, a 2-millimeter animal is resetting. Revising the terms. Beginning again as if the previous version of itself had never happened, renegotiating the only contract evolution has ever enforced without exception. We haven't found the strangest thing in the ocean yet.